Hi, my name is Trilotma and I'm in my third year of PhD in Alicia Rogers' lab in the Department of Biology. I will talk about gene regulation and how small RNA pathways regulate expression of genes in the germline of model organism C. elegans. Gene regulation is important for development and fertility of an organism, and it also protects the genome from foreign particles and environmental stressors. In hermaphroditic C. elegans, RNAi mechanisms coordinate proper gametic expression, wherein spermatogenesis is restricted at the L4 stage, and as the animal grows into adulthood, oogenesis gene expression manifests. To ensure this precise coordination during developmental timeframes, we hypothesize that RNAi pathways employ autoregulation and feedback mechanisms to fine-tune and maintain optimal gametic gene expression, in this case, spermatogenesis. An RNAi pathway called ALG34 RNAi pathway is central to spermatogenesis. In this pathway, argonaute proteins ALG3 and ALG4, along with 26 gRNAs, provide effector function to their target transcripts, followed by downstream amplification of 22G sRNAs by EGO1, which is an RNA-dependent RNA polymerase. This ALG34 pathway is independent of mutator-focused 22G RNA amplification, and yet we see in our study that mutator focus influences ALG34 pathway, manifesting as a genetic switch to control spermatogenesis. To understand this feedback genetic switch, we leverage MUT16 mutants which have disrupted mutator focus, causing a hindrance in the amplification of 22G small RNAs. For our methods, we use bleaching to synchronize wild-type and mutant worms. We performed RNA extraction using trizol and isopropanol. For mRNA sequencing, we used Dynabead kits and performed next-generation Illumina sequencing. Our Western blots were performed using anti-flag tags and secondary antibody anti-mouse IgG antibodies. We did fluorescence imaging by fixing worms onto slides with chilled methanol and imaged them on axiomagular microscope in our lab. Let's look at some results. Figure 1 shows brood size assay which revealed that MUT16 mutants under heat stress have drastically reduced brood size with immediate effect on spermatogenesis. Next, we performed mRNA sequencing to see sperm gene mRNA expression, which is downregulated in MUT16 hermaphrodites at L4 stage, as shown in the blue dots here. In the background, since we established that ALG34 RNAi pathway is central to spermatogenesis, we wanted to test for the mRNA expression of ALG3 and ALG4 and did reverse transcriptase qPCR, which showed downregulation of ALG3 and ALG4 in MUT16 mutants under heat stress at the L4 stage. This correlated with our Western blot and fluorescence imaging data, where we do not see the foci of ALG3 and ALG4 proteins at L4 stage under heat stress, and see depleted proteins in our Western blots for ALG3 and ALG4. To this end, we wanted to examine if this downregulation of ALG3 and ALG4 is small RNA dependent or not. So with small RNA sequencing data, we show that ALG3 and ALG4 are targeted by other small RNAs. This is interesting because this serves as preliminary evidence that small RNA pathways regulate each other. To summarize, we see downregulation of sperm genes, and the pathway that is central to spermatogenesis is also downregulated in the additive effect of MUT16 mutation and heat stress. So, despite ALG34 RNAi pathway being independent of the mutator complex, it is being influenced by it, wherein mutator complex dependent small RNA pathways act as a genetic switch to turn on the expression of ALG3 and ALG4 at L4 stage and turn off the expression of ALG3 and ALG4 during the adult stage. This ensures a smooth transition from spermatogenesis to oogenesis during development and ultimately maintaining optimal fertility of the animal. My next steps are to identify the small RNA classes that target ALG3 and ALG4 and to dissect the mechanism of feedback pathways in small RNAs. This study will provide us with a roadmap into the molecular interactions of RNAi pathways. This will allow us to further identify the mechanisms of heritable diseases and fertility issues and impact therapeutic implications in the long run. Thank you.